Welcome back, everyone, to our afternoon session, where the main focus is going to be on MSA research. I'm Pam Bauer. I'm Secretary of the MSA Coalition Board, and I'm also the co-chair of our research committee. Before we get underway, we just have a little announcement about the handouts. We have an extra handout from the first speaker. So Judy's going around handing that out, one per family, because we have a limited number. I just want to tell you about our research grant program, which is really exciting. This year was the first time that the MSA Coalition, or any nonprofit, has granted funds to MSA research specifically. Just to give you some detail about that, we had a great increase in our donations in the last couple of years. And because of that, we were able to make a decision that we were actually going to fund research. Prior to that, we were specifically providing support and information through our helpline and things like that, our website. But this past year, we were able to grant 7 seed grants to researchers. And our goal for this research is very clear. We want to find a treatment or a cure for MSA. Our scientific advisory board, which is made up of some of the preeminent MSA researchers in the world, they review each one of these grant proposals that we receive, and they rank them on their relative merit towards this goal of treatment or cure. Every proposal that we've received so far has been worthy of funding, but we're limited in the amount of funds that we have available to give. We funded 7 projects in the past year, but we had 10 additional projects that we had absolutely no money for. There are ideas out there. There are great researchers with great minds who are working on this problem. We need to find them funds. Our policy is to fund MSA research that happens anywhere in the world. There's great work now going on in Austria, France, Germany, and the UK, as well as here in the US. A breakthrough in MSA research could come really from anywhere. The amount we gave to each of these projects was small. These are what we call seed grants. The amounts range from $25,000 to $50,000 each, for a grand total of, and I'm so thrilled about this, $250,000. And with your help, we can increase that every year from now on. So without further ado, I want to introduce our first speaker on the subject of research. Dr. Vic Khurana is on the faculty of Harvard Medical School and Mass General, and is currently a visiting scientist at the Whitehead Institute for Biomedical Research. He's now working on a very exciting project that was funded by the MSA Coalition. I'll let him explain it to you in much more detail because I couldn't even hope to get close to it. Please help me welcome Dr. Vic Khurana.
Thanks so much, Pam, and thank you and Judy and others for the invitation. It's wonderful to be here. It's a real privilege to be here. So I'm going to give you a little update on some of the work that we've been doing over the last few years and how I think it might relate to MSA. I have a great deal of commitment to this disease because I see many patients in our clinic with this disease. We follow over 100 patients at the moment with MSA at Mass General, and we see those in a number of our clinics. And so it would be a tremendous privilege to be able to contribute to treating this disease that I've been becoming all too familiar with. So the work I'm going to tell you about relates to stem cells. And it's an exciting time in this field. I think it's important that we don't overpromise, and I think it's important that we do the work correctly. But I do think it's a very exciting time, and I'm going to make an argument that this is the right time for us to band together and to create a national stem cell registry for this disease. And I'm going to take you through step by step, and I hope you agree with me at the end and that we can discuss it later in question time. So for a start, what is a stem cell? There are many different types of stem cells. I think the stem cell that most people are familiar with from popular work is the embryonic stem cell. The embryonic stem cell is capable of forming any tissue in the body. So when a sperm meets an egg, you get a group of cells that are called totipotent. They can become any tissue of the placenta and a growing human being. But when these start to develop, an inner group of cells here in the fetus, just before you get to a fetal stage or in an embryonic stage, an inner group of cells can become any cell of the body. They can't become a placenta, but they can become any tissue. And we call those cells pluripotent, and they're known as embryonic stem cells. So one of the controversial aspects of stem cell research is, of course, that embryonic stem cell research involves embryos, human embryos, and everyone has different views about embryonic research. And that's why it was a tremendous breakthrough in 2007 when a Japanese researcher named Shinya Yamanaka really published a paper that I think is going to transform medicine. He won the Nobel Prize for this discovery in 2012. And what Dr. Yamanaka showed was that we could take an adult patient and take a biopsy, for example, of skin. We could grow skin cells from that patient, and we could turn them in a way that was far easier than anyone until that point had ever imagined into a cell that was very much like an embryonic stem cell. And so this is a cell that doesn't require an embryo. It actually just requires us to have access to a tiny little piece of skin, even a hair follicle, and we can now generate what are called induced pluripotent stem cells, or iPS cells, from any patient or caregiver. Anyone in this room could give a small biopsy and have stem cells generated. Now, I'm not going to get into the issues of embryonic stem cell versus induced pluripotent stem cell. Suffice it to say, it's my responsibility as a scientist to say that the only reason we know how to do anything with these cells right now is because of the tremendous research that has been done on embryonic stem cells, and we need, in my view, both of those aspects of research to continue for us to be able to move the field forward. Nevertheless, this was a tremendous advance for brain diseases because for the first time in history, we could take a patient who had a brain disease and we could generate their brain cells while the patient was still alive. And that's why it was such a tremendous advance. And so it involves taking a skin biopsy, making skin cells and turning them into these iPS cells, and those iPS cells can then generate any tissue in the body that you would want to study from that patient. And so one of the areas that people think about a lot and that you'll read about a lot is stem cell transplantation for neurologic diseases, and in particular in newspapers you'll read a lot about stem cell transplantation for Parkinson's disease. And because people with neurologic diseases tend to hear about the common diseases, I thought it was important for me to at least at the outset address some of what people are talking about in the context of Parkinson's disease. So Parkinson's disease, as you know, has some overlapping features with multiple system atrophy, particularly the MSA-P type of the illness. And the aspects of Parkinson's disease that are characteristic relate to slowness, rigidity, tremor, and instability of posture. And there's a loss of dopamine-secreting neurons, and importantly, many patients with Parkinson's disease, as you may know, respond to treatments with dopamine, drugs like Sinemet. And so one of the features of Parkinson's disease that really relates to that loss of dopamine is centers around a part of the brain called the midbrain, where there's a group of cells that secrete this, what's called a neurotransmitter, this chemical dopamine. And so you can replace this and you can get some symptomatic relief for a number of years for patients with Parkinson's disease. And some patients with MSA, as you know, also respond to this treatment, but much fewer with MSA respond to this. And part of the secret in Parkinson's disease is that while it affects many cells of the body, and the brain, I should say, it does affect in a fairly discrete way this area of the midbrain, which makes it responsive to dopamine. And so you can see here a normal patient. This is brain where the substantia nigra, which means the black substance, has these dopamine neurons, and it loses its pigment in this disease as these neurons are lost. And so one of the promises of stem cell transplantation is that we could now make from these iPS cells that I was telling you about, we can make dopamine neurons. And indeed, I and many others make dopamine neurons in the lab now, and the protocols we use are based upon some of the work that was done in Lawrence Studer's lab in New York. And so this is brought back research history. If you think back a number of years back to the time where cells from fetuses actually were put back into humans and showing some promise in Parkinson's patients. So that of course was controversial for a couple of reasons. First, it was cells that came from a human fetus, which was very controversial, as you would understand. Second, many of those patients developed severe complications of the transplantation, which was part of the humbling aspects of being a doctor and being a patient, that many of the things that we think are going to work in the lab are more challenging when it comes to putting them into patients. And so there were a number of things that stopped those fetal cell trials, but the advent of iPS cells has brought that research back into focus very clearly, because now we can take patient cells, make them into iPS cells, make dopamine neurons, and theoretically put those dopamine neurons back into patients with the disease. And I show you that in a schematic, right here, where you would take a biopsy from a patient, make skin cells, iPS cells, the nerve cells, the neurons, and you could transplant them back. So this is a promising area of research, and it should be pursued. There is one very important caveat, probably a number, but one that I'm going to discuss about this kind of transplantation research. And that is that Parkinson's disease, and more so MSA, do not just involve the degeneration of one group of nerve cells in the brain. Particularly MSA, but also Parkinson's disease, involve the degeneration of a number of groups of cells. And so just putting a certain group of cells back into the brain, even if it were to work with all of the potential problems that could happen there, even if it were to work, has its limitations, because these diseases involve much more than just the loss of a single group of cells. So that is true for Parkinson's disease. So this was the midbrain. This is taking a slice across the brain where you're seeing the brain in a kind of side-on view here. This is the top of the brain here. The eyes would be here. And this is the brainstem leading down to the spinal cord below. I'll point it out over here as well. And so I point out the area of the midbrain that we've focused on for Parkinson's disease, but Parkinson's disease involves many other parts of the brain too. And when you think about MSA, you're dealing with more parts of the brain still. As you know, MSA-C takes its name because it affects a part of the brain at the back known as the cerebellum, which is indicated here, which would not be, for example, responsive to replacing dopamine neurons. And as you would expect, many patients with MSA don't respond to dopamine because there's a lot more going on in the brains than just the loss of those particular cells, even though that does happen in MSA. So there is an issue with taking one group of cells and putting it back for this disease that I think is a fundamental one. And so there have been two approaches that you'll read about that I'm happy to answer questions about. I'm going to talk very briefly about one and much more about another, the one that we're pursuing. The first is that if you can't just put back a single group of nerve cells into the brain, maybe you could put back a stem cell that secretes growth factors that would actually rejuvenate parts of the brain, maybe not reverse the disease, but actually provide growth factors that encourage the health of the surviving nerve cells that are still in the brain. And so there's been some interest lately, and many of you might have heard, so I thought I would address this up front. There's been some interest in what are called mesenchymal stem cells. These are a type of stem cell that originate that you can bring out of the bone marrow, and they grow into cells that are not brain cells. They grow into muscle, they grow into fat cells, they grow into cartilage, bone, but they have some very interesting properties in that they secrete growth factors that may be very important for regenerating or for nourishing brain cells. And so there's certainly a good deal of work that's happened and is happening in this area. And so it's on your handout, but what I'm basically saying in this slide is that there are these cells called MSC, mesenchymal stem cells, and they can not only become certain cells, but they can secrete various factors that can help neuronal health. They can help the insulation that goes around the nerves, and also for encouraging nerve growth. So theoretically, they're an interesting cell type because they don't replace the cell that's being lost, but they might help cells that are still in the brain. And so a group in Korea has performed a number of studies with mesenchymal stem cells, and the more recent one in 2012 was a blinded study. It was reasonably well done. And they showed some effects, actually, some promising effects. So if you look at this graph here, now the way they did this was they injected these they took someone's bone marrow, they grew these cells, and they injected these mesenchymal stem cells back into the blood of those patients. They did it for once a month for a few months, and then they followed the patients for a year. And if you look at this graph, the people who didn't get the cells are on the top, and the people who did get the cells are on the bottom. And the higher the number, the more the motor effects of multiple system atrophy. And so over a year, these patients did look like they were doing better, the ones who had received the treatment versus those who did not. And the scans below, on the left, you see a scan that shows loss of the top part of the little bit of thinning of the top part of the brain that we know as the cortex that happens in this disease also. And you can see the red color is for more progression, and the lighter yellow color is for less progression. And so the patients who received the stem cells on the right had less progression of the thinning of tissue in their brain. And so this was a very interesting study, and something that really needs to be pursued. There are a number of really important caveats or concerns with this research that I wanted to bring up. The first is we don't really know how these cells are actually working. We're injecting them in a kind of blind way into the blood of a patient, and we're seeing some kind of effect, but we don't know how it's working. And whenever we don't know the mechanism, we have to be careful, because a lot of the time when we don't know the mechanism of something, it turns out over time those kinds of treatments look promising in the beginning, but they don't end up panning out. That doesn't mean they shouldn't be pursued. They absolutely should be, but we need to learn more about what these cells are doing. We need to have further studies. There needs to be multi-center trials, and they need to be carefully done. The other question here is safety. So one of the, and to their credit, one of the studies that this group did was they looked at MRIs of the brain of these patients, and they showed that after the injection, they were getting very small stroke-like changes in the brain. So there were little parts of the brain that were not getting the right blood supply after the injection of these cells. And that's a serious concern because you wouldn't want patients to have a serious stroke, for example, and other effects. And we don't really know what the long-term effects of this treatment are. We also don't know how long any given injection of these cells lasts. And so while I'm enthusiastic about this work, I would say it's very early stage and it needs a good deal more understanding of the biology before we can advocate for this kind of treatment. So then there's a second approach which I'm going to focus on a little bit more. And the take-home message for the kind of early research that I do is that I think that to treat any of these diseases, whether it's Parkinson's or MSA, we're going to really need to understand their mechanisms. Because as much as we all want to give treatments, we've seen time and time again that when we rush this process, we not only have things that don't work, we spend a lot of time and resources on them, but we also set the field back because people like yourselves start to think that nothing's going to work, and there's a loss of hope. So I think we need to attack the problem of MSA at different angles, and we all need to get together and do things differently. But I do think we need to, at different levels, understand this disease better. And so one of the efforts and the way in which we're looking at this problem really was born out of work that was done in the late 1990s. And this was the finding, and a really seminal finding for MSA, that there was a particular protein that was aggregating, that was clogging up cells in the brain, not just nerve cells, but cells called glial cells, oligodendrocytes. These are cells that provide the insulation around if you think of our nerve cells as having cables, these are cells that provide the insulation around those cables. And each of these cells puts cables around many nerve cells. So you can imagine if you start taking one of these cells out, you're taking out 100 nerve cells, for example, if you take out one of these support cells. So these are really critical cells in the brain. And it turns out there was a particular protein called SYNUCLEIN that was aggregating and causing problems in this cell, in the brains of patients with MSA. And so MSA until that time had many different medical names, and actually it was thought to be a number of different diseases. But it was the finding that there was this one pathology in the brains of these patients that brought the field of those different diseases together. And one of the most critical aspects of this is that we already knew about this protein because this is the same protein that aggregates and causes problems in Parkinson's disease. And that, I think, is a tremendously helpful finding for the MSA field, because what it means is that therapies that are directed at this protein alpha-synuclein, which is involved in Parkinson's disease, may well have implications for other diseases in which this protein aggregates, including MSA. And in fact, MSA, the MSA patient population may be an ideal one in which to test synuclein blocking treatments that are now starting to emerge from Parkinson's disease research. So our research has really tried to target this particular protein, synuclein. And it's a longer road. We have to understand what this protein does and how it affects the cells. But we think that by creating a logic against this protein, we will understand this disease better and be able to get treatments to patients. And so here's a slide on the left of brain tissue with Parkinson's disease, and on the right side with MSA. And what I'm showing with the arrows are nerve cells that are having accumulations in brown of this protein alpha-synuclein, and it's accumulating in Parkinson's disease on the left and in MSA on the right. And so it is the culprit protein. And how do we know it's the culprit protein and not just something that's happening in the brain that's not related to the disease? Well, it turns out there are a small group of patients who have mutations, a single change in the genetic sequence of this gene, synuclein, and they get very early degenerative changes in their brain that leads to Parkinson's disease, it leads to dementia, and it leads to aspects of the disease like autonomic dysfunction that you heard about this morning that are related to MSA. And so we know that a single change in the genome, in the genetic code the billions of letters that spell out our genetic code. A single change in this gene is capable of producing the kind of pathology that we see in these diseases. So we know it's an important protein. And so now the approach that we're taking is, rather than transplanting cells back, we are growing these cells and growing nerve cells from our patients, and we're using them for drug discovery so that we can figure out how this protein synuclein is causing problems in brain cells and how we can fix it, how we can reverse it. And so this is the idea of having Parkinson's disease, or MSA, literally in a dish where we can grow these cells. It's not the same, of course, as a patient's brain, but on the other hand, it's the first time we've ever been able to have these cells for us to study from living patients. So we think it's an exciting time. And the idea here is these are the cells, for example, the dopamine neurons I was talking about from a patient who has accumulation of alpha-synuclein. These are cortical neurons, which are in the, as I say, the top part of the brain, the cortex, which is involved in our cognition and our memory and our thinking. So we can now look at these. We've always been able to look at these from brain tissue after a patient has passed, but now we can grow these cells in a dish. The same kinds of cells who have the same kinds of features, we can grow them, and we can watch as they go from an embryonic stem cell-like state into a neuron. We can watch the progression of a disease. So when we started this work about 6 years ago, there were two really big problems we were facing. The first was, how do we actually find abnormalities in a dish for a disease that might take decades to develop? So we're growing these cells in a matter of weeks. How are we going to find those earliest problems in the dish that relate to these diseases that, as you know, take a long time for them to show up in life? So that was the first problem. And the second one was, who was the control? And by that I mean, if I was to take a skin biopsy from everyone in this room, we all have different genes, our cells would all grow differently. How would I know what the critical changes were between one person's cells and another that were capturing what was really important about that disease? Because everyone's cells in this room are going to be different. And so there are going to be many differences that have nothing to do with disease. And how are we going to capture the actual problems that really relate to the disease? So these were the two really fundamental problems that we were facing in the field at that time. And I'll show you one of the approaches that we took. The first was, we found, remember I told you that a single change in the genetic code of that gene synuclein can cause this disease? So we found an exceptionally rare patient who had one of those single changes. And she was a very generous woman, she's passed now, who flew to Boston and gave us a sample of her skin about 6 years ago. And what we did was we, in what was a very difficult effort and a collaboration with another group, with Rudolf Janish's group at MIT and Frank Soldner in his lab, what we did was we took those cells and we edited the genome. And by that I mean, we changed that mistake in the synuclein gene, we changed it back to a normal synuclein code. And so what we can now do, and it's a little sci-fi, so bear with me, but what we can now do is we can make stem cells from patients and we can genetically edit them to change their genetic code. And so we knew that there was a single change in this person's genome that was causing this disease, so all we did was we changed it back. And what that did was it allowed us to compare this woman's cells to her own cells, but one copy of the cells had the problem gene and the other copy had the fixed gene. And that means all of the rest of the genes in the dish were the same. So we didn't need to worry about them. So just as I was saying, if I took everyone's cells here, they'd all be different. Well, we didn't have that problem anymore because there was only, out of the billions of letters in the genetic code, there was only one difference between these cells. in that gene, alpha-synuclein. And so that allowed us to start this field. So that allowed us to really get going with this aspect of understanding these stem cells in the dish, because we knew that these cells were identical except for that change. And that's how we started. So we created, so what you're seeing here on the left is we created these embryonic stem cell-like cells from the patient, and we created our own cells that looked exactly the same for all intention and purposes, that looked the same. We created the same cells with that mutation corrected, okay? And so we had these brain cells now on the left that had the disease, and then we had the cells on the right. I've just pseudo-colored this. They, of course, didn't look different color. But they looked identical, really, and as you would expect, this is a disease that takes some time to develop. So now we had these neurons. We knew we would have to compare them, but we were still left with the problem, where do we start? Where do we start with this? This was a completely unknown, we were in really uncharted territory, I should say, because it's not like the cells instantly die. They don't die if they've got the abnormality. We know that because this patient lived till her 50s, and so she was quite able to live with these cells. So it wasn't something sudden that happened, so how are we going to find it? So we needed a way to be unbiased, and this is another thing that I want to emphasize today. We all have different views about how these diseases start, and some of those views come from looking at postmortem brain. We have different views about, whether it's related to oxidative stress, and an antioxidant might help, or a vitamin, and we all have different views about it. But we wanted to start in a way that was completely unbiased. We did not want to go in with a preconceived idea of how this protein was causing its problems. But we couldn't do that. It takes months and months to grow these cells. We couldn't screen these cells for hundreds and thousands of compounds. We couldn't chop and change every gene in the genetic code because it would take 100 years and $1 billion to do it, and too many people. So we had to start in a simple way. And so I'll tell you what we did. So the idea was, in a dish, could we find a method to change the disease cells back into something that looked like the disease-free cells? Could we find drugs that could do it? Could we find genes that do it? So all of you have heard of bread, and all of you have heard of beer. And one of the key features of bread and beer are these little tiny cells called yeast, okay? And yeast cells are a rather remarkable cell, because as much as you would think they were a simple cell, they're actually rather complicated, and they have all of the core machinery that every one of our cells in the body has. So if you think about cancer research, and if you think about many other aspects of human disease research, it turns out that a lot of what we know about how a human cell works is from studies that were done in yeast. And the reason is that we can take this simple cell, and over the last 30 or 40 years, we have developed methods to chop and change every gene in its genetic code. And we can screen it not against hundreds of drugs or thousands. We can screen this little organism against millions of drugs. And so what we did was we used a platform that had been developed at MIT by Susan Lindquist several years before, where she had put the protein alpha-synuclein into yeast cells. And it causes, just as it causes all sorts of problems to happen in nerve cells and brain cells, it really caused a lot of problems in yeast cells too. And not only that, it caused the same kinds of problems that we knew were happening in brain cells. And so I can elaborate on this a lot more later if you like, but suffice it to say it was a very interesting model system which was capturing some of the key pathologies we knew were happening in the brain. And so what we did was we utilized data from Susan's lab, where she had screened every gene in the genetic code of this cell and found out in an unbiased way what were the proteins that made the cell better, and what were the small molecules or things that could be drugs later, what were the things that could make those proteins better. And so in other words, what were genes and drugs that we could find in this simple cell that could actually fix the problem of this rogue protein, alpha-synuclein. And so it turns out we found a number of those genes, And they led us down particular pathways. They led us to look for certain things in the brain cells we had made from this woman. And sure enough, we found abnormalities that were very similar, that were coming out in a completely unbiased way from this very simple cell. Not only that, we found small molecules, things that could become drugs, that could reverse those deficits and change the diseased neurons back into something that looked like the healthy neuron. So we created a logic around this protein, alpha-synuclein, where we could understand what it was doing to a cell and we could understand how to fix it. And it's only the beginning of the story, of course, but it's an important logical step. And so over the last few years, and I should say, this slide actually takes us one step back in terms of why we would do this in yeast. If you're going to do something crazy like this, you better be sure that there's really a good reason to do it, right? And so on the left here, interestingly, are blockbuster drugs. Now, the top one is a drug that I'd vouch. There are a number of patients and people in this room on this medication. These are called statins. Statin drugs, things like Zocor and Lipitor, control cholesterol. Well, it turns out that a statin hits the same gene in a yeast cell as it hits in a human. And that's true for many of these blockbuster drugs here on the left. They hit exactly the same gene in a yeast cell as they hit in a human. So there's a lot of evidence for drugs that we know, very well and that we use, that if they had come out of studies in yeast, they would have actually worked in people, because we know that there's a lot of things that work, we know work from before, that actually work in the same way in these simple cells. And so that was part of the justification. Another interesting aspect that you might not be aware of is if you look at the entire tree of life, well, it turns out fungi like yeast are right next to animals like us, which is a little humbling. But that just draws attention to what I said before, that the actual machinery in one of these simple cells is very, very similar to the cells that we have. And so over the last 5 years, as I've mentioned, we've found genes, small molecules, and now a drug that I'm very excited to tell you about, that can reverse synuclein toxicity. So what do I mean by genes and small molecules? I mean these are not synuclein. These are other genes in the human genome or in the yeast genome that, when you put them back into the cell, can make them better. But that's really difficult to get to the clinic. Small molecules are one really important step forward. These are compounds that could become drugs, but as you know, the process of drug development takes a long time. And so these are very promising, and they are about to be going through a drug discovery and a drug development process, which we're very excited about. But a drug is something that's already been used in patients. And it's very desirable for us to find things that we know already work, because those are the kinds of things we can get to patients sooner. And so in the last year, we have, and this is work that's been spearheaded by one of my colleague, Gabriella, have found that a particular drug, FK506, which is an immunosuppressant drug that's been used for 20 years, can actually reverse the changes of alpha-synuclein. And it reverses it not just in these simple yeast cells, it reverses it in patient neurons, so neurons that we develop from patients, and it also reverses the problem in rats that we've engineered to express more of that protein alpha-synuclein. So we've done a number of trials in rats. This is still very preliminary data. More trials need to be done. But what's really interesting and exciting is that we know how this drug works, and we know how it affects the biology of this rogue protein alpha-synuclein, and we know how to exploit those facts to get it to be therapeutic, at least in cells and in rats. And so there is a tremendous amount of excitement now around this, because we can start to think about, dream about putting this into patients. And I'll talk a little bit more about that in a minute. But this has been a team effort, of course, and so Susan has been a pivotal part of this effort, developing the yeast platform. I developed the neuronal platform with my wife, Chian. and worked very closely with Dan Tardif and Gabriela Caraveo at MIT to do this work. And so just a little bit more about FK506. It was discovered in 1984. It was FDA approved in 1994 for immunosuppression after liver transplants. It's now used in kidney, heart, small bowel, pancreas, you name it, transplants. So we have tremendous clinical experience with it. It readily crosses the blood-brain barrier, and we know it's a lot about how it gets bioavailable, how it crosses that barrier and gets into the brain. And what's important, of course, from the point of view of MSA is that it reverses alpha-synuclein toxicity from yeast cells to rats to Parkinson patient neurons through a mechanism that we know. So through a logic that we have uncovered now and that we are working more on. And so I would put it to you that this is certainly early work, but it's the kind of work I think I believe in strongly because it creates an understanding of how one builds a program to get to a patient. So you start in a very simple cell, you then go into an enormously complicated cell, a brain cell from a patient, but now we have to make the leap between these complicated cells to patients themselves. And that's a challenge, and it's going to need a lot of resources, but we think we've set up a logic that we can, that's exciting and worth pursuing. In particular, now I want to just talk about the implications for MSA directly. And so as Pam mentioned, I was a very fortunate recipient of a seed grant for some work on MSA specifically. I care a lot about this disease. I see a number of patients and a growing number of patients with this disease. And as I say, for me, one of the real rays of hope here is that we think we know the protein that is really heavily involved in this disease process. So if we can understand how this protein does the damage and understand how to fix it, I think we can make tremendous strides. And because it's the same protein that's involved in a very common disease, Parkinson's disease, we can piggyback in some ways on the Parkinson's community so that we can all work together to really fight this protein, because I think that's what it's coming down to from my point of view. And so what the seed grant has allowed me to do is start to create patient iPS cell lines, so these stem cell lines from my patients with this disease. And not only that, some of these patients have passed, and I'm tremendously grateful that some of them have donated their brains to Mass General so that we have their stem cells and their brains. And it's a difficult thing to raise, but it is tremendously important for the stem cell field that we understand the abnormalities in these cells. And I can't tell you how grateful I am to those patients for that generosity, because it's one way for us to validate the tools and techniques we are now developing so that we can help other patients with this disease. And so what you see down here is one of our first patients who gave her skin. We made the skin cells, and then they change over time in the dish, and then you can see over time they grow into these colonies here that have been stained in green that are iPS cell colonies. So we are now growing different cell types from the brains of these patients. And so the two big kind of pathways for me forward for this disease, one is really related to generating stem cells from patients, as I've mentioned, so that we can identify defects, so that we can engage in therapeutic testing. So I've mentioned FK506, the drug that we're working on, but there's also that Korean group that's working on mesenchymal stem cells. And remember I said we don't know how they work? Well, by capturing patient cells and neurons and identifying their abnormalities in a dish, we can actually start to figure out what are the factors, what are the things coming out of those other stem cells that were potentially making these cells better. So we can start to understand how those cells that were being injected in the blood of those patients were actually helping them. Because if we find the mechanism, that will lead to, I think, opening up a therapeutic approach for the disease that's different to the drug-related approach that we're pursuing. Biomarkers. So as I said, we can watch these cells grow in the dish over time, and we can start to get clues about what changes over time. That's a critical thing for us to take back to patients with MSA, because if we don't know what's changing over time in patients, we won't know that a drug going into a patient is doing its job. So we need to create a way to track this disease in time, and stem cells that we can now grow from patients may be an important way. And finally, of course, we can use these cells for drug screening. But one of the issues here, and it's a good issue and a bad issue, the good side is that MSA is not a disease that directly runs through families, as most of you know. And that's obviously a good thing. But one of the things that makes it difficult from the research point of view is I can't take an MSA patient's cells and do that genetic correction that we did for that other patient who had that SYNUCLEIN mutation. And that means we're faced with the same problem that I started with. How do we know that what we're seeing is really relevant to the protein alpha-synuclein and really relevant to a patient's disease? So it turns out that those patients with synuclein abnormalities that we have grown cells from are being highly informative for our research in MSA patients. So that I think is tremendously promising. So we are starting, and this is very preliminary, we're starting to identify defects in these cells related, we think, to the protein synuclein, which I think is very promising. But it's going to need a lot more work. But we're also going to need strength in numbers. We need to understand this disease better. We need to know if it's one disease or many diseases. We need to be able to have a bank of these cells from different parts of the country, from different patients with different clinical characteristics, to really start to get at this problem, because this is a complicated disease where we're not going to be able to do those genetic fixes. So that's one of the aspects that I really want to emphasize. And the second is, what are we going to do now with this drug that we have, FK506? Well, of course, we would love to offer it to patients. But before we offer it in a therapeutic sense, we need to be able to figure out in living patients if it's doing in living patients what we think it's meant to be doing in the dish. And so we need to, again, extend the logic of what we've found in patient cells. We need to extend that to patients. And so we are now thinking very much about how to establish a biomarker program in Parkinson's and in MSA where we can start to look at the pathways we know are affected by this drug and see if those change over time in patients and see whether they are engaged by the drug when we give it to patients. So before we even think about treating symptoms, we again want to be very clear on how this drug is working in living patients. And so it's an exciting time where we can start to make these plans, and I want to engage the MSA community in this. We can start to make these plans to get this drug into patients. And as I've said, the justification for a stem cell bank is really strength in numbers. MSA is a so-called sporadic disease. It's not a genetic disease, which means we cannot correct it genetically in a dish. And I've said, I've asked the question just a few minutes ago: is it one disease or many diseases? We need to be able to address this issue. And the message I'm really going to leave you with, if there's one statement that I think really covers what I do and what my approach is and the approach I'm advocating for here, is that we're going to really need to understand it in order to treat it. And so I am just tremendously grateful to be here, engaged with you as a community, and I want to engage with other clinicians and other scientists so that we can all think about different avenues to understand this disease and to treat it. So with that, I thank you very much.
Thanks very much, Dr. Khurana. And on behalf of everyone, I'm going to give him a big hug because I think this is wonderful. We're going to hold questions until the very end. We're going to have a panel up here at the end to answer all your questions.